So we're going to find we have something called natural killer cells. Did y'all know that? They're pretty cool, okay? They'll help us with virus-infected cells and cancer cells. And that becomes pretty cool. Inflammation. Inflammation, believe it or not, can be a way that the body tries to fight off a pathogen. It can actually be beneficial for something to get inflamed. I know, go figure, right? Phagocytosis. We're going to have cells in this first and second line of defense that want to eat the stuff up. Fever. How many of you ever thought that or knew that the fever is needed?
Oh, yeah.
But the minute we get a fever, what do? We're going to try to get that fever down. You only need to get it down if it's about 100. If it goes over 100, start worrying about it. Other than that, don't worry about it. Seriously, just say it. Okay, so what is some of the stuff that we can get exposed to that the body can work against under this first and second line of defense? Well, if we look at something, like I said, that's going to be species resistance, this is where we may not have a receptor for a particular pathogen that we might get exposed to, and therefore we're not going to develop it. Okay? We're not going to develop something that we might have gotten. Now, good example. Some humans are resistant to malaria. Why? They've got sickle cell. Now, there's a lot of controversy over it, okay? But they believe that because of the sickle cell rates in the area of the world that's high in malaria, that this was actually an evolutionary adaptation. There's a lot of controversy over it, okay? In my mind, I can see how that could be the case, okay? Because people just kind of need to get over that term evolution. You know, you tend to denote a lot of negative stuff with that. You need to kind of push that in the background, pay attention to what is being said. Now, another good example, humans, we get stuff such as measles, syphilis, gonorrhea. Do other animals get that kind of stuff? No. Well, cats can have if you're thinking about like the feline AIDS, okay? It's not the same. Now, characteristics of it are similar between what happens with the cats and what happens with the humans, but that can't cross. So in other words, the cat can't give it to the human and the human can't give it to the cat. Yeah, so it doesn't cross the species. So we actually have the ability to have this development of a resistance in a species as a whole. Mechanical barriers. We have our skin and our mucous membranes. Think about those mucous membranes. Do you happen to remember what they line?
Openings.
Openings to the exterior of the body. The mucous membranes are very important in our first and second lines of defense. The skin covers the body as a whole, and that's a huge first line of defense for the skin. if it makes it past whatever that pathogen might be, if it makes it past the skin, if it makes it past that mucous membrane, then we have to move into our second line of defense. So you kind of seeing what I mean by first and second lines of defense? And it doesn't matter, okay? If something makes it past what we think of as the first, all right, the body's going to react the same no matter what, all right? The chemical barriers, now they get a little more interesting. For the chemical barriers, one of the chemical barriers that we have, well, under our enzyme family, now think about this. What is it? What is an enzyme?
Biological catalyst.
Well, it acts as a catalyst to help speed up a reaction, but what is it made of?
Protein.
It's a protein. And proteins are what? What are they made of? What makes up a protein? Amino acids strung together, having their primary, secondary, tertiary, quaternary structure. Okay? So, what codes for the structure of those amino acids? Your DNA. Okay? The genes on your chromosomes. So, in this enzyme family for immunity, our body, have you guys ever heard of an interferon?
Yeah.
Why? How?
Cancer. Using medication.
You've probably heard the term, but haven't probably ever really sat down and thought about them, okay? But they're actually very important in the body, and especially against stuff like viruses, okay? So interferons...
Isn't there a medicine you take for cancer that's an interferon?
Mhm.
Okay.
Yep, and they have like injections, like if you've had exposure to a certain pathogen, they can give you injections of it to help basically boost that immune system. Interferons, they're hormone-like peptides. Now look at what they do. They stimulate phagocytosis. You guys remember monocytes, for example? Okay? So they're going to help to stimulate cells that can carry out phagocytosis. If they can stimulate cells that can carry out phagocytosis, this means the cells, they don't really care what it is. If they just recognize that it's a pathogen, they go and try to eat it up and kill it. Defensins. Somebody was really inventive in lab that day, okay? Defensins. Once again, peptides. Look what produces them. They're produced by the white blood cells. They basically are a chemical that when they come into contact with the microbe, kill it. Collectants. These can help with certain bacteria, yeast, viruses. Now, complement. Your textbook is going to get very detailed about complement. And note the spelling for this. It's not like going and saying, oh, you look so nice today. I love your top and look at your hair. Ooh, okay. Not that type of compliment. That's C-O-M-P-L-I. This is C-O-M-P-L-E. Complement, group of proteins. Your textbook is going to get very detailed about these. Okay, I don't feel like we really need to get that detailed about them, okay? But look at what they do. They're a group of proteins in plasma, body fluids, And then look what happens with complement. Stimulates. What did I say about inflammation?
It can be good sometimes.
That it can be good sometimes. What's this doing?
Except
Bring it into cells that'll eat, and then enhance it. They're important, all right? But what will happen? Complement this group of proteins. If we activate a group of the complement, that'll activate another group of complement, which will activate another group of complement. And basically it'll end up being a cascade reaction. So if we end up getting a cascade reaction, we're activating more of it, which will just activate more, which will just activate more to carry out the job that it's doing. Does that make sense? Your book will get really detailed about C1, C2, C3, okay? I'm just not going to get into that with you. All right, does that make sense? Natural killer cells. I just like the name. I like that somebody got inventive that day in lab, okay? A small population of lymphocytes. Let's stop it right there, okay? Small population of lymphocytes against viruses and cancer. And what do they do? Secrete cytolytic substances. Stop right there. Cytolytic. Take that term apart for me. Do what, Jessica? Cell lyse. So it's telling me they will lyse the cells. They do this with something called perforins. What does that make you think about? Huh? To perforate. And if something is perforated, this is perforated, right? Okay? Got holes. Yeah, this is pretty cool because what it'll do, if there is a virus or a cancer cell that it's going to attack that cell that it's trying to kill, it'll come up, release those perforins. The perforins create holes in the cell. Water from the body will rush in and the cell burst. Cytolytic. Is that not the coolest? Do you know that that's how our very first antibiotic functioned? What was our very first antibiotic? Penicillin. Do you happen to remember when it came out?
I thought it was like 19.
It was the early 1900s. Guess who it was first tested on? Which the majority of stuff is tested on anyway. Huh?
It was kid, wasn't it?
Well, as a group, as a whole populace, like trying to get it out into the populace.
Did it rabbit or mouse? Doctors. No.
Our military.
Oh.
You'd be shocked at what they do to our military, you know? And came out, World War II is when it began to be used on that population. And the reason they were using it is because in World War I, we had more deaths from typhoid and stuff like that, okay? And they found out, wow, penicillin's pretty good. And that's the only way penicillin worked. It would go up to the bacteria, punch holes in it, water from our body would flow into it and burst the cell. I just think that is so cool. But that's just me, okay?